Good morning or afternoon or whenever you're watching this. This is our day one learning on viruses. Today we're going to learn just about viruses in general. Tomorrow we're going to learn a little bit more specifically about our friend SARS-CoV-2, which is the virus that causes COVID-19 caused by SARS-CoV-2 is a coronavirus, and that's kind of what everybody refers to it as. But hopefully after the next couple of days, you're going to learn exactly what's going on with viruses and be able to speak intelligently about what the heck is going on in the world today. Those viruses are just interesting organisms to take a look at, even though they're not alive. So let's dig in. You are going to want to take handwritten notes on this because you'll have a quick quiz coming up in a couple of days. So make sure you're writing down things and using those notes only on your quick quiz. Comes up. All right, let's go. So what are viruses? They are protein coats surrounding either DNA or RNA, and you get a couple ribosomes in there to make proteins. So what does that structure actually look like? They have three basic structures. You have the helical structure right here. That's mainly plant viruses that look like that. And you've got, this is the one that we're all familiar with, right? Many, many shots on TV. This is a circular virus with these little spikes sticking out of it, and we'll talk more about that later. That's forming the corona shape, hence the name coronavirus. Most human viruses and animal viruses look like this. And this guy at the bottom is a bacteriophage. And so what's cool about him is that he only infects bacteria. So we don't really talk about him that much at all because he only infects bacteria. We're interested mainly about this guy right here, right? So what do viruses do? Where's their name coming from? The name comes from a Latin term that means slimy, liquid, or poison, which is kind of, we can relate to that right now, right? And viruses are not alive. You know this. We've talked about this before. What does it mean to be alive? You can reproduce, you grow, you replicate, you carry on metabolic activities, all that kind of good stuff. But the thing with a virus is that even if they do all those things, and they do, they have to have a host cell in which to do those. So viruses are not considered to be alive, which makes it very difficult to actually kill them. There comes my cat. And henceforth, we have to kind of attack what they're made up of. So we attack this protein coat around the outside here. We can maybe attack these little guys right here, these little spikes that help them to attach to ourselves. We can attack this interior metabolic activity of the virus, not allowing it to replicate once it's inside the cells. We can do a lot of those things. But first of all, let's talk about a little bit more about the structure of each one of these little guys. So the first one, this is electron micrograph right here, and that's a picture of, like, it's a cylindrical shaped virus, and it's just a little helical shape. So it's a little spiral of proteins. That's all these things are made up of. These are proteins, and this is mainly a plant virus that we're looking at here. The next one, this is our friend that we're familiar with. This has got a protein coat, aka a capsid, and then it has these little spike proteins sticking out of the protein coat, and they help that virus to attach to living cells. It has DNA inside. Some of the viruses, like our friend the SARS-CoV-2, has RNA instead of DNA as a genetic material. And last but not least, we've got this little weird alien-looking guy. He is a bacteriophage, and his sole purpose in life is not to attack animal or plant cells, but only to attach itself to bacteria so that the bacteria can do the work of replicating and making virus babies, which is kind of weird and creepy. It does look like an alien, that's for sure. Next, we have just the proof that viruses are not alive, because no matter what we do to them, if they're outside of a living host, they will never carry on metabolic activities. So they do have the DNA or RNA that's necessary to make copies of themselves, but they have to have a living cell in order to do all of those things. So where did these weirdo-looking things even come from? They could be pieces of DNA and RNA that escaped from plants, animals, other kinds of cells. And interestingly enough, there are fun facts down here at the bottom throughout this entire lecture. Of course, we have to keep things interesting for you. So if you talk about a milliliter, right? So a little milliliter is a really small amount. We've measured that before in our labs. And milliliter in each milliliter of ocean water, we have 10 to the 30th power of virins or viruses. And if you lined all those bad boys up, I think the bottom part is cut off here, but it's 200 million light years into space, into all the viruses strung together that are just in the ocean, which is kind of crazy, I think. All right, let's keep going here. Next, we have a fun fact for you. But before we start our fun fact, we have those 3 domains that we talked about. Remember, we've got the eukarya, the prokarya, and the archaebacteria, right? So we are eukaryotes, and prokaryotes are our bacterial friends. They are alive. And then archaebacteria is this weird, crazy domain filled with bacteria that live in extreme environments, like in the vent of the deep ocean, near a volcano, or in the hot springs, or things like that. So those are the 3 domains. And viruses, even though they're not alive, they do evolve because they do have DNA or RNA inside of them. So what's interesting about that is that they are kind of classified like living organisms. They all have their own names and things like that. They're not alive. So where did they come from? Not quite sure. Little pieces that might have evolved and then formed this protein coat around themselves. No one actually knows. What makes viruses kind of cool. So what we're going to look at here is this parasitic wasp. Oops. And we're going to, if I can get this to work, we're going to get our friend to show you how some viruses hijack the bodies of other organisms. And in this case, the virus is laid in the eggs of a wasp into a caterpillar's body, which is really crazy to think about, right? So the virus is inside the eggs. The wasp lands on the caterpillar, injects her eggs into this poor little helpless caterpillar, this little guy up here. And then the eggs will grow and develop, and then they have to come out of the caterpillar. Now, the weird thing about this virus is that it does some funky things to the caterpillar once the eggs grow and develop. Let's see what we've got
Our parasitized caterpillar has spent the last 12 days gorging itself. It now appears profoundly obese. But this
eggs are inside the
is
caterpillar.
not all fat. The Glomerata wasp larvae lie just under its skin. Each is the size of a grain of rice, but together they account for over 1/3 of the caterpillar's weight. The larvae have not yet finished growing and need to keep their host alive. So although they feast on the caterpillar's blood, they have been careful not to touch a single one of its vital organs. This uneasy truce will not last. Within days, the larvae are fully matured. Suddenly, they begin to stir into action. For the past 2 weeks, this surrogate womb has protected them, but now they no longer need it. To complete the next stage of their life cycle,
inside the
they
caterpillar.
must break out. The caterpillar's thick skin should be a solid barrier to the parasitic wasp larvae. But as their bodies have grown, they have developed tiny, saw-like teeth. These jagged jaws are for one job only: cutting their way out. Stroke by stroke, the larvae slice through the tough layers of skin. At the same time, they release chemicals that paralyze the caterpillar. As the larvae break through, there is nothing it can do. Free at last, the larvae enter a new phase of development. They swiftly spin silken cocoons. These will provide the perfect environment for their final transformation. But ironically, one of the greatest dangers the larvae will face is being themselves impregnated by other species of parasitic wasp. Incredibly, the wounded caterpillar helps them out.
This is where the virus comes in.
Usually, a caterpillar would spin a silken blanket to make its own cocoon. But the parasitized caterpillar spins his blanket on top of the wasp cocoons, giving them an extra layer of protection. Scientists believe the same wasp virus that infected it weeks before has now invaded the caterpillar's brain and caused this bizarre corruption of its normal behavior. Amazingly, the caterpillar's natural aggression is now also exploited by the wasp virus. The caterpillar becomes a bodyguard, actively protecting the cocoons from other parasites. It will watch over them unceasingly until it eventually starves to death.
Okay. That was pretty crazy, right? So a virus that was laid in the eggs of this wasp inside this poor little caterpillar up here takes over the entire brain of the poor caterpillar and causes it to have that crazy behavior. First it's paralyzed so that the little the larvae can eat their way out. And then it's still alive and it protects the eggs, weaves its own cocoon over them, and then stands guard. That's crazy, right? Totally nuts. All right, let's keep going. So how do viruses reproduce if they're not alive? Well, there are three basic ways that they can do this. One is to attach themselves to a host. They inject the DNA or the RNA inside of the living cell, or they are encapsulated inside of the living cell, and all that cell does is make virus babies until it explodes, killing the host, right? Temperate viruses are what most of us are familiar with, and they are the ones that we get infected by. They hang out for a while, we fight them off, and then they can go into hibernation sometimes, or we can just completely get rid of them and fight them off. So some viruses, like the herpes virus, you never get rid of it. It's always lurking in your body, which is kind of strange and creepy. It actually likes to live, the herpes virus likes to live at the base of our spines and hang out. And then it will come forth usually during times of stress. So that's a temperate virus for you. And then a lysogenic reproductive cycle is when the DNA or RNA is incorporated into the host's DNA or RNA, and then it's making the virus proteins and basically making new virus babies right along with all of its other jobs, which is kind of a weird thing also to think about, but that's what's happening. All right, so in the first one, in the lytic cycle, you have 5 steps. So we talked a little bit about the protein spikes that were sticking out there. A lot of times those are used to attach to the cycle or to the cell. And then you have to penetrate into the cell membrane, usually cell wall also, if it's bacteria or plant cell. And then you're going to inject the DNA or RNA into the new cell. You have to replicate that so that you're making more copies, more copies, more copies. And then you're assembling the new virus parts. So you're assembling the new capsid, and then you have to release that from the host cell. That's part of the lytic cycle. So what does that look like? In the case of our bacteriophage friends, here's what they do. So these little alien little guys, they will attach to the cell wall. Remember, bacterial cells have cell walls. They'll attach to the cell wall of the bacterium, and they're going to attach, inject their DNA or RNA, whatever it is, and then the DNA is going to code for things. What does DNA code for? Proteins and other things. When the DNA starts to code for those proteins that are being made, it's saying, hey, I need to make the bacterial capsid, the viral capsid that's going to make our new viruses for us. So we're going to replicate that. And then once I've made all of those replications of all those little virus parts, I've assembled my complete virus down here, and then that bacteriophage is released from the bacterial cell, and it then goes to invade other bacterial cells. So that's how bacteriophages reproduce. That's what it looks like. Don't they look like aliens, right? So that's what bacteriophages look like. Attaching to a cell, you can see they've lit it up with nuclear material. Very exciting. And so we're watching the DNA being injected into this bacterial cell. They really do look like space invaders, don't they? So in the lysogenic cycle, this is the one, remember, that doesn't kill the host cell because it's not bursting wide open with virus babies. So you have the same thing that happens. So you've got to attach to the cell, not always bacterial cells. This can be any type of cell, but it's just showing it in a bacterial form. And then the DNA is incorporated right here into the DNA of the cell there. And when it does that, it's going to cause the cell to, whatever kind of cell it is to make those proteins along with what they also already make. So it's not going to necessarily kill the cell. It doesn't kill the cell, but it's going to reproduce all of that particular DNA right here that's been incorporated into the genome of the new cell. Now here we go. What we've got with our friend here. Let's go up here so you can see this part. And face out of the way. There we go. So in the virus that we're looking at now, this coronavirus, and let me get my drawing capabilities here. So we've got this guy going on, and he's floating around in the bloodstream, right? He's just looking for a cell. And in the case of our friend SARS-CoV-2, he happens to like our lung cells a lot. So that's where it wants to go and to hang out. It's got the perfect conditions for it to do its dastardly job, which isn't evil per se, because it has no brain to think of things like that. All it wants to do is replicate itself. That's its sole purpose. So what does it do? It uses these spikes that are on the outside, capsid here, to use them to attach to these receptor proteins on the membrane of the cell. So our lung cells then have these receptors so that the virus can attach to them. Once it attaches, then it can fuse. Oops, I didn't mean to do that. Okay, I don't know what I just did, but we're going to figure it out. So it's going to fuse with the membrane of the cell, and then the virus is now inside of the cell. The capsid has to be dissolved using enzymes, and then the DNA of the cell, there we go, the DNA of the virus is going to be incorporated into the cell's DNA right here. The DNA from the virus comes into the nucleus where the DNA of the cell resides. So let's pretend that this is our DNA in the nucleus of our cells. And it's going to cause the same thing to happen, protein synthesis, that the DNA does for any of our proteins that need to be made, right? So you have the DNA, you have to, I'm going to draw again for us here. The DNA is going to be copied using a messenger RNA. Remember that during protein synthesis. And so once the messenger RNA leaves the nucleus, it's going to find the ribosomes. The ribosomes are where protein is made. So I'm going to make my new proteins right here in the endoplasmic reticulum. Remember, that's going to go through there and get processed and all that kind of good stuff. And the different kinds of proteins, you can have the proteins that are embedded in the capsid, you can have the capsid itself. All of that is being made. And these proteins that are on the outside of the virus, see how they're now being formed so that they are incorporated. There we go. So there we go. They're being incorporated into the membrane of the cell. And so that means that it can recognize now future viruses that attach to it, right? So we've got our virus that's being made here, and it's going to be leaving the cell and going to invade other places in the body. I'm going to bring this down for us. There we go. Okay. And so now we see the entire cycle right here of the virus that we're looking at now, where it's got to attach. DNA is, and the entire virus itself can be encapsulated by the membrane, brought inside. It has to be dissolved. The capsid has to be dissolved. The DNA is going to go in the nucleus. Just like any other DNA that needs to be read, the messenger RNA is going to make a copy of that. It's going to go out, find a ribosome, either attached to the endoplasmic reticulum or not. The proteins that make up the new virus are then created. And we've got the proteins here that are making up outside of the virus, right? So then this cell can now recognize new viruses that are being produced. And then the virus, the new virus that's itself being made with the new DNA and the new capsid is going to be enveloped and incorporated into the membrane, pinched off, and now I have a new virus, including the spikes that are on the outside. So it's kind of a complicated process for making a virus. It sounds like it's a complicated process, but it's really not once you learn the steps of it. All right, so viral diseases. Now we get to the fun stuff, right? Like the craziness of what do these viruses do. They usually aren't very friendly, meaning that they don't help us out a whole lot. Sometimes they can become vectors for us and get information like DNA to certain places in the body. But most of the time, they're going to cause a disease or at least some symptoms. So we've got a couple of diseases that can be caused by either DNA or RNA viruses. So DNA viruses just have DNA on the inside. RNA viruses have RNA on the inside. That's really the main thing. So the particular virus, the coronavirus, ooh, nice writing here. That is an RNA virus. That means it has RNA inside of it. A lot of our other viruses that we battle with are DNA viruses, and so they just have DNA inside of them. In the case of rubella, it's a form of measles. You got a vaccine for this when you were little, probably. It is an RNA virus, and it causes a fever, and you got this nice little rash, usually pretty itchy, and then it tends to go away. Once you've had a disease like that, though, you always have parts of the virus inside of your body. So remember back in the day, H1N1, that was in 2009, we had over 61,000 cases confirmed in the United States alone. And you'll notice that, boom, 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 looks very familiar to what you've been seeing on the TV. It's got these spikes that stick out of its protein coat. And we were getting vaccines and through the nose, and they weren't very fun here. You can tell this poor little guy's having a good day. But people tended to freak out a little bit about H1N1. Some schools were closed, very few though. It was very an isolated case, and then we got a hold of it, and then all was well. So there you go, nice and neat for you right there. All right. Next, we have our new friend, SARS-CoV-2, right? So that's the one that everybody's all worried about. It causes COVID-19, and it's an RNA virus, just like the measles. We're going to do a little bit more work with this guy tomorrow, but wanted to show you again the structure of it is something that you already seen. It's the circular spherical shape right here, and it's got these proteins that are embedded in them. We'll call them the protein spikes, and we call it corona because it has this crown shape of these proteins around it on the outside. So plant viruses, most of them are RNA viruses. My husband is dancing off screen and trying to make me laugh, but I'm not going to do it. And how are they spread? How are plant viruses spread? Mainly through vectors like insects. A vector is something that transports the material from one place to another. And we can also spread them through the plasmodesmata, or the connections between the plant cells within the cells themselves. Most of the time, they are this helical shape, and they can do things like create tobacco mosaic virus is one virus in particular that can wipe out an entire crop. So they do kind of the same things in plants that they do in animals and humans, where they can create this cascade of problems, sometimes killing the host. Right? What are retroviruses? HIV, our friend, we haven't really talked about that in quite a while, because there was a big uproar in the 80s and 90s. And now that we kind of have a handle on this particular virus, we know how it works, we know how to treat it, we know how to slow it down, and kind of almost put it in hibernation, so to speak. So what is HIV? It's a retrovirus. So what does that mean? That we've got an RNA inside of it, and we're going to take that RNA and make it into DNA that is then incorporated into the DNA of the host. So it stays in the host cells basically forever. Hepatitis B is also a retrovirus. All right, a couple of random facts for us here. We're going to take a little break and see if we can get this to move up for us. Are we going to do that? Sure we can. Okay. So the first flu virus that we kind of know about is the virus of 1918. We didn't know a whole lot about it at the time. We knew it wasn't bacterial in nature, but it was so small we had a hard time realizing what exactly it was. So in 1933, scientists said, oh, this is a weird thing. It's not bacteria. It's this other kind of a thing. And so then we went back and we figured out that the 1918-1919 pandemic, which killed millions of people, was what we call the Spanish flu virus. HIV is a small little virus, meaning that its genome is only about 10,000 nucleotides long. The smallest virus that we know about is called the circovirus, and they're very small. They only have 1,700 nucleotides. Nucleotides are those A's, C's, T's, and G's, remember, that make up the genomic sequence. We also have some super viruses. They're called mini-viruses, kind of a cute name, right? And they can be over 1 million nucleotides in length. And they are very complex, and they code for lots of different proteins. So what is this protein capsid? What is it made up of? And why are our viruses so difficult to destroy? Well, the reason why they're difficult to destroy is that outside of the body, we can just bleach the heck out of them. We can use soap, and soap breaks down a lot of these coats, right? And bleach breaks down the coats. Last time I checked, soap and bleach were not things you want to inject into your body. So then how do we attack these little guys? What do we do? How do we destroy them, right? So first of all, we've got these spikes right here. Let me draw again for us. So we've got these spikes. They're on the outside, and they're embedded into the capsid, and they allow them to latch on. And if we can block the spike, we can block the attachment, then that's a good thing. But if we can't, then that's the problem that we're being forced with. Also, number two, that the viruses aren't alive. And because we have a difficulty destroying the protein code itself, what makes up this whole thing, we have difficulty destroying the virus. However, if we could block that, if we could block the targeting on the cells, and so these guys could not attach, then that would be a really good day. And that's what we're actually trying to work on here as scientists. We being the scientists all over the world are working on this very thing. So how do we destroy these guys? How do we keep them from replicating? How do we break down their protein code? That's what we're looking at right now, which is kind of a cool thing. So viruses throughout history, we're going to take a brief walk through some of the big famous pandemics that have been recorded in recorded history. And so far, things are unfolding, of course, but the COVID-19 disease has been less deadly than the annual flu virus. We're going to see how that unfolds and see if the numbers grow with that. But we're watching it because it's so easily transmitted. Tomorrow, why this particular virus is so easily transmitted. But here are the sources I got where I used to find all this information about our worst pandemics. So let's take a look there. First one is the HIV virus that causes AIDS. We're going to go from most recent to the most distant past. And this is the most recent one that really caused a lot of chaos. Ask your parents about this one when it was publicized in the probably late 80s is when we first really started hearing about it on a large basis. People started to freak out because it was 100% fatal. Gosh, you get this virus, it destroys your immune system. It stands for human immunodeficiency virus because it wipes out your immune system. All those great white blood cells that attack the invaders in our bodies, bacterial cells and viruses, it shuts that whole thing down. So when you get HIV, you become infected with the virus, it can become AIDS. What we're doing now is once someone is HIV positive, we can kind of block that virus from taking over or hijacking the immune system and shutting it down so it doesn't become full-blown AIDS. But once you do have AIDS, basically a cold will kill you because you have no way to fight off any kind of virus, any kind of bacterial infection, anything at all. It just kind of shuts your body down. So we first identified it way back in 1976, which is interesting. But it didn't really come to the forefront until the late 80s because it started killing people in great numbers, 36 million since 1981. We first kind of saw it here in the United States and we started paying attention to it globally. All right, next one, we've got the 1968 Hong Kong flu, killed 1 million people. All right, we may think, well, globally that's not huge, but think about it. We're back in 1968 and we're starting to become a little bit more global. So you're noticing the structure of these viruses that I'm showing you. It's going to probably come up as a little pattern in your brain. We're going to talk more about that tomorrow. These are coronaviruses, which is a general overall classification of viruses. So the mortality rate of this particular virus was only 0.5%, but we still were paying attention to it globally. We wanted to start tracking these things because we had better technology. Most of the people who died were in Hong Kong. It's the origin of the disease. And 15% of the population. That's a pretty big number. If you took 15% of the United States, that's several million people. So remember, this is back in 1968. And scientists have been tracking that. The Asian flu, a little bit earlier back in time, so about another decade earlier, you're noticing the structure of the virus again. Yep, circular with these little spikes. It's going to become a theme here among these huge pandemics. It is also caused by a flu-type virus, killed about 2 million people. And what does this H2N2 and H1N1 even mean? We're going to talk that tomorrow. But suffice to say that it was another kind of an Asian area type flu. We think to ourselves, well, oh my gosh, what's going on over in China and Asia in general? What's going on? Why are they having so many flu bugs? Well, if you ever were to visit China, you would notice that the big cities are pretty much where most of the population lives, and they live in very small, close quarters. Usually they're living in apartment buildings and they're doing their business interactions very close in the marketplaces and things like that. And so diseases tend to spread easier in cities. So that's why we're seeing a lot of these Asian viruses. Spanish flu is the famous one, the 1918 Spanish flu. Here's our friend down here at the bottom, remember, with the spikes sticking out of him. And this one killed folks between 20 and 50 million. Oh my gosh, that's a huge margin. Why do we not know the numbers? Well, because no interweb, right? We don't have a way of recording it. Many of the people who died were in rural areas and there was no way to diagnose this at all. And so what happened is that they tried to document as best they could, but we're talking 1918, 1919, right? Still pretty early as far as recording things down in an accurate manner. And it would strike down anybody. Kids, middle-aged people, teenagers, older people. It didn't matter. It killed without looking at any kind of age group. So now we've got, these particular age groups that are vulnerable. The Spanish flu, it didn't care. You either fought it off or you didn't. And interestingly enough, if you look down here at the bottom, how did they treat it? The best way was to roll those patients outside and let them get fresh air. Sounds familiar, right? Fresh air and sunlight are the best ways to treat this particular virus. So yes, we had the whole thing with the face mask and all that kind of thing. So many people were affected, and at that time we had zero clue how to treat the flu. None at all. And so people were either, like I said, they either were strong enough to fight it off, they created the antibodies and then they were able to fight it off, or they didn't. All right, then we go back into the 1800s. It's a little crazy now. This was the type of flu. And by the way, we kept bodies of people who died in the 1918-1919 pandemic. They were found in like permafrost, so their families would bury them. And in much colder climates in certain areas of the world, there are a couple areas in the world where they found enough of remains of people way back when that they were able to isolate that virus and study it. It's kind of weird. So we're looking actually at the 1918 virus structure now today, because in the last couple of decades, we were able to find these people who had died, and enough of their bodies had been preserved so that we can get the virus out. Kind of weird, but kind of good also, because then we can look at the structure, we can compare it to viruses that we're dealing with today and things like that. So the flu epidemic, or pandemic, in the late 1800s killed about 1 million people. We called this one the Asiatic flu or the Russian flu. Again, we're looking at sources outside of the United States, but people were infected also here. Again, we're looking at the H2N2 or H3N8. We're going to talk tomorrow about what those letters mean. But it was also a big killer. 1 million people is a lot of people. So what about the plague? Everybody says, oh my gosh, the Black Plague. It killed 75 to 200 million people. That was a global rocker back then, because look at the time frame, folks. 1346 to 1353. Wow. Okay, so I'm going to draw a little bit more on here. What does this mean? The Black Plague was bacterial in nature. So our friend, the bacteria here, this little bacterial guy, he could be killed by antibiotics. So if we had had antibiotics back in the 1300s, we could have really saved millions and millions of people. But we didn't. And so, unfortunately, we lost a lot of people because we didn't have the technology that we have today. So the plague is caused by the Black Plague, or the Great Plague, or the Black Death, caused by a simple bacterium called Yersinia pestis. Kind of an interesting name, right? And so how did it get transmitted to humans? Creepily enough, it was transmitted, my husband is correct, rats, fleas on rats, to be specific. Fleas on rats were the cause of the spread of the Black Plague. And if you look at the time frame, the 1300s, it was not a very good time for hygiene. And so this disease spread rapidly through populations. And by the way, this is the reason, this Black Plague is the reason why we bury people 6 feet underground. Because they discovered that if you buried a body just a foot or two under the ground, say it was really cold outside and the ground was frozen, and you were mourning, you didn't have time to really bury that person, unfortunately, the rats were going to get to the body and they were going to become infected. And then the fleas were going to become infected that lived on the rats and then pass that along. So the 6 feet underground comes from the Black Plague. And that is it for today, my friends. We will talk more tomorrow about our friend, the COVID-19 disease, and we will also talk about how it spreads and what we can do. Until then, have a great day.